In this video, we're going to look at the structure of DNA. Thanks to all the research that had been done beforehand, knowing that DNA was indeed the genetic material, a lot of research in the mid-1950s was conducted to actually look at the structure of DNA. Let's look at the individual nucleotide structure itself. Before we can study DNA, we need to look at each of the nucleotides to understand what the structures are. There are three very important parts to every single nucleotide. When I talk about nucleotides, I'm referring to adenine, thymine, guanine, and cytosine. Number one, there's a phosphate group, and that phosphate group makes up part of the very important sugar phosphate backbone of DNA. Number two, there is a pentose sugar. In the case of deoxyribonucleic acid, or DNA, that pentose sugar is called deoxyribose sugar. The carbons on that sugar are labeled as 1', 2', 3', 4', and 5'. In this study, we will focus mainly on the third carbon, which is the 3' carbon, and the 5' carbon in the deoxyribose sugar. Thirdly, we have the nitrogenous base itself, the adenine, the thymine, the cytosine, or the guanine. When we look at the nitrogenous bases, really we break them up into two subcategories, the purines and the pyrimidines. Let's look at the purines first. They're easy to recognize because they always have a 6-membered ring full of nitrogen attached to a 5-membered ring. Guanine and adenine are both considered to be purines. When you look at the pyrimidines, on the other hand, they only consist of a single 6-membered ring, again, full of nitrogen. That's why we call these the nitrogenous bases. The three pyrimidines that we find in our nucleic acids are cytosine, uracil, which remember is only in RNA, and thymine. There's an easy way to remember the purines from the pyrimidines, and here it is. Biology is pure agony. The purines are adenine and guanine. On the other hand, you cut a pie. The pyrimidines are cytosine, uracil, and thymine. Well, in the 1940s, a researcher by the name of Erwin Chargaff conducted some really important studies. He looked at the DNA from many organisms, and he compared it. We're going to take a look at some of this data. The Homo sapien, of course, is a human. We're going to look at the fruit fly, otherwise known as Drosophila melanogaster. In addition to that, he studied the common bread mold that we've looked at earlier and E. coli. When Chargaff studied the amount of adenine and thymine in each of these organisms, he found that the A's almost equaled the T's. So in the human, you can see that the concentration of A and T are very closely related. However, in the fruit fly, A and T were similar, but were different from the A and T concentrations in the human. Likewise, in the common bacteria E. coli, they tended to have the same concentration roughly of A and T, but their concentrations differed from the other two organisms. The same was true when you compared cytosine and guanine. Cytosine was always equal to guanine, relatively speaking, within a species, but clearly there were differences when you compared one species to another. So, in conclusion, adenine is equal to thymine, and cytosine is equal to guanine. This gives rise to Erwin Chargaff's famous rules. Number one, the amount of A, T, C, and G in DNA varies from species to species. Human DNA, for example, is more AT-rich than bread mold or E. coli. It turns out that's partly due to the fact that A and T are more easily broken when DNA replicates. Ironically, this wasn't figured out till a little bit later. Number 2, in each species, the amount of adenine is always equal to the amount of thymine. Next, when you look at the concentration of A plus C, that's equal to T plus G. Interestingly enough, we have a purine here and a pyrimidine here. That's also equal to a purine here and a pyrimidine here. Those two, no matter what species we're looking at, represent about 50% each. Next came two very important scientists in the study of the DNA structure. Perhaps one of the most influential female physicists of the last century, or even in history for that matters, is Rosalind Franklin. Rosalind Franklin, along with her counterpart, Maurice Wilkins, conducted X-ray crystallography of DNA, and their studies produced some very interesting data that laid the way for Watson and Crick, who came very soon after. Their studies showed that there's a possibility that DNA is indeed a helix. Well, in order to confirm that, Watson and Crick had to take this data and use it to produce their very important model. 1953. This is where James Watson, the US researcher, coupled with Francis Crick, the Great Britain, when they came together in Great Britain to build these Tinker Toy-like models. Believe it or not, some of those models were over 10 meters tall. They were made of wood and plastic, and they built those models based on Wilkins and Franklin's data, and they came up with some very interesting results. In the end, they figured out that one complete turn of the molecule represents 10 base pairs. Very interesting. And furthermore, their measurements showed that that distance represents about 3.4 nanometers of distance. Not a big number. Remember that a nanometer is only a billionth of a meter. They also determined, thanks in large part due to Erwin Chargaff's results, that guanine is triple hydrogen bonded to cytosine, and of course adenine is double hydrogen bonded to thymine. So indeed, the sugar phosphate backbone is there to hold the rungs in the ladder, the A, T, and GC bonds that exist in DNA. This model over here kind of unpacks the double helix in a little bit more detail. So it's quite easy to see that adenine and thymine join together with the physical bond between the nitrogenous bases. Meanwhile, a triple hydrogen bond is present between guanine and cytosine. Also, really important research found by Watson and Crick involved this whole concept between the 3' end of the molecule and the 5' end. According to Watson and Crick, DNA was not just a double helix, but it was also anti-parallel. And anti-parallel means that each rung in the ladder runs in different directions. And I'll explain this in a little more detail. When you look at the 3' end of this left portion of the deoxyribose sugar, it runs upwards towards the 5' end, which included the phosphate end of the last sugar on the left side. When you look at the right side of the molecule, ironically, the 3' end starts at the top and works downwards towards the 5' at the bottom. This may not mean a lot to you right now, but it turns out that the anti-parallel nature of DNA, the fact that it runs 5' to 3' in one direction, and 5' to 3' in the opposite direction is what explains its ability to replicate itself. So, in conclusion, we've unpacked a lot of knowledge here. DNA is definitely a double helix thanks to Watson and Crick. We know that it contains a sugar phosphate backbone. The 4 nitrogenous bases form the rungs in the ladder that keep it together via those physical bonds known as hydrogen bonds. Adenine is double hydrogen bonded to thymine, and guanine is triple hydrogen bonded to cytosine. DNA is complementary base paired. We know that adenine must be bonded to thymine, and we know that cytosine must be based with guanine. If that doesn't happen, it turns out later that we've got ourselves a mutation. Fourthly, we know, according to Watson and Crick, that DNA is an anti-parallel molecule. That means that there's a phosphate end on the 5' end and then a hydroxide end on the 3' end, and DNA runs in 2 directions. Another picture here shows the same story. We have an anti-parallel molecule. This DNA molecule here runs from 3' to 5' on the left side and 3' in the downward direction to the 5' on the right side. I hope this short video helps you understand the basic structure of DNA. It now paves the way, really, for us to figure out how this molecule has to divide prior to mitosis. Well, it turns out that replication of DNA is critical only because of the hydrogen bonds between adenine and thymine and the 3 hydrogen bonds between cytosine and guanine.